Please.
Okay. Good morning to all. A warm and heartfelt welcome to each and every one of you joining us for today's enlightening webinar. I am Dr. Sanjay Nagdev, the co-convener of this event, and it brings me immense pleasure to witness such a diverse and engaged audience gathered virtually. Your presence not only enhances the vibrancy of our discussions, but also signifies a shared commitment to pursuit of knowledge. I eagerly anticipate an insightful and collaborative session as we embark on this intellectual journey together. Thank you for being a part of this enriching experience. Today is a significant milestone as we are gathered here for the 38th SPSR webinar on SiRNAs, a new era in cancer chemoprevention, coinciding with the World Cancer Day. This event is part of a distinguished series hosted by the Society of Pharmaceutical Sciences and Research under the esteemed mentorship of Dr. Shashi Alok Sir, National President of SPSR. Under his guidance, the SPSR continues to play a pivotal role in advancing knowledge and innovation in the field of pharmaceutical sciences. The SPSR is dedicated to advancing the pharmacy profession through a multifaceted approach. Our objective encompasses the organization of conferences to campaign the causes of pharmacy, contributing to programs and services that address the health needs of India and focus on disease prevention. Additionally, we actively recognize and honor distinguished pharmacists who contribute significantly to the encouragement of the pharmacy profession. At the core of our mission is the promotion and facilitation of the acquisition, dissemination, and exchange of pharmacy knowledge, ensuring a robust flow of information about the practice of pharmacy and pharmaceutical services. Through these objectives, we aim to elevate the standards and impact of pharmacy profession, fostering a dynamic and collaborative environment. We are truly honored to welcome esteemed speaker Professor Gaurav Kaithwas sir, Dean School of Pharmaceutical Sciences, Babasaheb Bhimrao Ambedkar University, Lucknow. Professor Kaithwas brings invaluable expertise to our event, and we eagerly anticipate gaining insights from his wealth of knowledge in the field. His presence enriches our discussion and adds significant value to our exploration of the latest advancements in the field of pharmaceutical sciences, particularly in the context of cancer prevention. We are grateful for his participation and look forward to a truly enlightening session. In our esteemed gathering, we are honored to have the distinguished presence of Mrs. Monica Sabarwal ma'am, the convener of this webinar, National Secretary of Society of Pharmaceutical Sciences and Research, and Managing Editor of IGSPSR and IJP Journal. Her multifaceted contribution and leadership in these capacities underscore her commitment to advancing the field of pharmaceutical sciences and research. We are privileged to have Mrs. Monica Sabarwal ma'am with us today, guiding our webinar with her expertise and dedication. And without further delay, I would like to invite Mrs. Monica Sabarwal ma'am to extend a warm welcome to our esteemed speaker, Professor Gaurav Kaithwas sir. Over to you, ma'am.
Thank you so much, Dr. Sanjay Nagdev. A very good morning, everyone. A warm welcome to the 38th SPSR webinar on SRINN, a new era in the cancer chemoprevention. I am Monica Sabarwal, the convener of this significant event, and it is truly an honor to have each one of you to join us today on the occasion of the World Cancer Day. On World Cancer Day, we unite in the global fight against the relentless adversary. Let us stand together, honoring survivors, remembering those we have lost, and dedicating ourselves to research, awareness, and support. Today, we reaffirm our commitment to the world without a cancer, where health trumps over the illness and hope conquer despair. Today, we can make strides in prevention, early detection, and treatment, fostering future where every individual lives are free from the grip of cancer. Our relentless pursuit of knowledge and advancement in the field of pharmaceutical science and research, the Society of Pharmaceutical Sciences Research, SPSR, takes a great pride in bringing together the experts and scholars from across the globe to explore groundbreaking topics that have a potential to revolutionize healthcare. Today, we gather to delve into the intricate world of SRI-NRAs and their transformative roles in the cancer chemoprevention. The SRI-NRAs, or the small interfering RNAs, represent a promising frontier in our ongoing battle against the cancer. This webinar serves as a platform to discuss the latest advancements, research findings, and the future prospects in harnessing the power of SRI-NRAs prevent and the combat cancer. We are privileged to have with us a distinguished speaker, a luminary in the field, Professor Gaurav Kaithwas, Dean of School of Pharmaceutical Sciences at Babasaheb Ambedkar University in Lucknow, Uttar Pradesh, India. Professor Kaithwas is a visionary researcher and educator whose expertise has significantly contributed to the advancements in the pharmaceutical sciences. We eagerly anticipate his insights and knowledge sharing on this very crucial subject. It is my immense honor for us to extend a heartfelt welcome to the Dr. Shashi Alu, the esteemed National President of SPSR. Dr. Shashi Alu's exponential leadership and commitment to the scientific research have played an important role in propelling our community forward and fostering a culture of innovation. Under his visionary guidance, has been instrumental in orchestrating a remarkable transformation within the SPSR, positioning it as a beacon of excellence in the field of pharmaceutical sciences. I would also like to express my sincere appreciation for the invaluable contribution of Dr. Sanjay Nagdev, co-convener of this webinar, and the organizing committee members, Dr. Saurabh Kose, Dr. Pradeep Golani, Dr. Sanjay Nagdev, Mr. Ankur Agarwal, and Mr. Junaid Tantre. Their tireless efforts have played a crucial role in transforming the vision of this event into a reality. Behind the scenes, there is an exceptional team who has been a backbone for the seamless organization of this webinar. Their tireless efforts and dedication, commitment to excellence have been a driving force behind the success of this event. I myself extend a gratitude to each and every member of SPSR team for their meticulous attention to detail and their passion which have made this webinar possible. The overwhelming response to this webinar is truly heartening. We have received more than 25 registrations from 16 countries, highlighting the global significance and collaborative spirit of our shared commitment to combat cancer. As we embark on this intellectual journey together, let us foster an environment of open dialogue, exchange ideas, and collective learning. Your presence today reflects your dedication to staying a forefront in the scientific innovation and making a meaningful impact of future on the cancer research. Once again, thank you for being part of the 38th SPSR webinar. And without further ado, let us begin this enriching exploration into the promising realm of SIRNAs and their potential to shape a new era for cancer chemoprevention. Do not forget to subscribe at the SPSR YouTube channel to stay updated on the latest webinar and research insights. Thank you, and let us begin the 38th SPSR webinar. Over to you, Dr. Sanjay Nagdev.
Thank you, ma'am, for allowing me to conduct this webinar. And now let's begin with a brief introduction to Professor Gaurav Kaithwas, sir. We are honored to have Professor Gaurav Kaithwas, sir, with us today, a distinguished professor in the field of pharmacology at the Department of Pharmaceutical Sciences, Babasaheb Bhimrao Ambedkar University, Lucknow. Professor Kaithwas had made significant contributions to the field of cancer biology with a primary focus on the development of small chemical entities to counteract cancer hypoxia. His impressive academic journey includes earning his undergraduate and postgraduate degrees from College of Pharmacy, University of Delhi, and the Department of Pharmacology, Jamia Hamdard, respectively, with a remarkable 127 publication, boasting an average impact factor of 4.14, along with three patents to its credit. Professor Kaithwas has established himself as a leading figure in the scientific community. His postdoctoral training at the Infectious Disease Laboratory, National Institute of Immunology, New Delhi, and Department of Biophysics, AIMS, New Delhi, further demonstrates his dedication to advancing knowledge in his field. Professor Kaithwas has secured extramural grants exceeding 3.23 crores from esteemed funding agencies such as DST, SERB, and ICMR. His outstanding contribution have been recognized with several awards, including the G.D. Bhale Rao Medal from the Zoological Society of India, the Young Scientist Award from the European Institute of Scientific Research on Ayurveda in the Netherlands, INSA Visiting Scientist Award, Best Researcher Award in 2014 and 19 from BBA University, NASI INSA Fellowship, Raman Fellowship, and many more. Beyond his accolades, Professor Kaithwas currently holds the office of Dean, School of Pharmaceutical Sciences, and Head of Department of Pharmaceutical Sciences at Babasaheb Bhimrao Ambedkar University, Lucknow. Additionally, he serves as the Associate Dean, Academic Affairs at the university. We are privileged to have Professor Gaurav Kaithwas, sir, as our esteemed speaker today, and we eagerly anticipate gaining insight from his wealth of expertise and accomplishments. It brings us immense joy and pride to share we have garnered the participation of over 2,500 registered attendees from across the globe, representing a diverse array of more than 12 countries. Among them, we have significant presence from India, Algeria, Kuwait, Nepal, Nigeria, Pakistan, Saudi Arabia, UAE, and UK. The overwhelming response from such a wide reaching and varied audience is truly humbling. We extend our heartfelt gratitude to each and every participant for choosing to be a part of this significant event. Thank you all for joining us today. Let make this webinar an insightful and enriching experience for everyone involved. We have our esteemed guest speaker, Professor Gaurav Kaithwas, sir, with us. Welcome, sir. Kindly, I request you to share your PowerPoint presentation and talk. Over to you, sir.
Thank you, Dr. Nade. Thank you, Dr. Shashi Alok. Thank you, Dr. Monica, for wonderful introduction. मतलब sometimes I felt कि मतलब मुझे भी नहीं पता था इतना सब कुछ मेरे बारे में. Anyhow, in any case, that I'm really thankful to the office of the SPSR for enabling me to interact with the fraternity associated with the SPSR. Dr. Nagdev, मेरी आवाज साफ आ रही है, क्लियर आ रही है?
Yes, sir. Yes, sir. एकदम clear, sir.
मेरी वीडियो भी क्लियर आ रही है?
Yes, sir. Sure.
Oh, I'm sharing the screen. If it is properly shared, then please let me know. Then we will start.
Yes, sir. Screen share हो रही sir आपकी.
I'm changing a slide, and please let me know whether it's changing or not.
Yes, sir. It's changing, sir.
Oh, okay. Thank you. So let us start with I will not take much about myself because Dr. Sanjay and Dr. Monica has already spelled a lot of about me. I'll just do a minor correction that as of now, I'm looking after the office of the Dean School of Pharmaceutical Sciences. So this is the title of my presentation. This is small interfering RNAs, siRNAs, which has emerged in last decade as a new era in the cancer prevention, chemoprevention. I'll be dividing my whole presentation will be divided into two phases. One will be I'll be talking about the basics of the cancer, okay, and chemotherapies associated with the cancer in very brief only. And then thereafter, I will be talking about the siRNAs. Then first, I will be talking about the RNAs, and thereafter, I will be discussing with you all about the small interfering RNAs and the RNA interference. We have been we know all that the cell is the basic structural and functional unit of life. So whenever there is a cell, whenever a cell is born in the body, it works, it performs its functions, and then it dies off. It is killed. Now, the killing of the cell is basically regulated by the process which is called as apoptosis. It is also called as a programmed cell death. Apart from the apoptosis, cells die because of the necrosis. Necrosis is like some sort of injury whenever it happens to our body, and due to the injury, the cell dies, and that is called as a necrosis. Apart from that, apart from apoptosis and necrosis, we have autophagy, pycnosis, and several other mechanisms which are programmed mechanisms of the cell death. And see, anyone who has taken a birth, who got the birth, either it is a cell or from cell, a group of cells, it is a tissue, from tissues to organ, and organ to organ system, and then to the body. See, whosoever has born, that has to die. Either it is in terms of humans or it is in terms of a cell. And the death could be injury based, like, or could be programmed, or the third thing is that the death might not happen. So it happens in case of cells that sometimes few chemicals, few viruses are there which actually induce some sort of mechanisms within the cells that the cell is not able to uh, hmm. I lost my screen. I'll just डॉक्टर संजय, मेरा स्क्रीन विजिबल है प्रॉपर्ली?
ये सर आपका स्क्रीन विजिबल है, बस आप स्लाइड शो मोड में बस कर दीजिए सर।
अब ठीक है?
हाँ सर, अब ठीक है सर।
Okay, thank you. So whenever any sort of a radiation, chemical, or sometimes some pathogens, majorly which are viruses, they induce some sort of changes in the basic structure, the basic genetics of the cell. And after that, what happens that the cell fails to die. The cells completely, it keeps on multiplying. And once it keeps on multiplying, it forms a different type of a cell. It becomes a different cells, and it generates and produces entirely different type of cells, which are constituently different from a normal cell. And such type of cells are called as a tumor cells. And whenever this uncontrolled growth happens, because those cells, they cannot die, they cannot go into the apoptosis, they cannot go into the autophagy or pycnosis. And necrosis, definitely it's a sort of injury, we cannot induce ourselves over there. So that forms a tumor, and tumor is an uncontrolled growth of a group of cells which follows the normal route of a cell, rule of a cell division. Now, how this formation and how this tumor cell formation happens, in a very brief, I will say that the tumor is a solid mass of a tissue that forms when abnormal cells group together. Suppose there was a one cell, if you can see on my slide, on my cursor, if suppose there was a one cell which was actually a defective cell, and it started multiplying. Once it has started multiplying, what will happen? It will grow and it will form a mass. It will form a organ system of a defective cells, and that is called as a tumor. In brief, tumors, they are of two types. One, our basic classification of the tumors are of two types, that is the benign tumors and the malignant tumors. Since majority of the listeners are from pharmacy backgrounds, I can feel that, I can understand that they will be understanding what is a benign and a malignant tumor. In brief, a benign tumor is some mass of a cell which actually remains confined to its original area and location. It is not going to invade into the other cell or other organ. Okay, by any means, it will be remain within the boundaries. And such type of benign tumors are easy to treat, because once ever it is found that there is a tumor mass, okay, but it has not invaded the other parts of the body, it can be simply removed from the body. Whereas whenever the benign tumor grows, whenever the benign tumors expands, and when it needs the energy, when it needs the oxygen, it needs more ATP, then what happens that it gives the signals for the metastasis, and subsequently the benign tumor is converted into a malignant form. What does it mean? It means that the tumor will be invading to the surrounding normal tissues, which process is called as a metastasis. And subsequently this spreading from the surrounding normal tissues goes throughout the body via the circulatory and lymphatic system, and such type of a tumor is called as a malignant tumor. In general etiology, if I just simply briefly try to understand what is the how the pathogenesis of the tumor starts, like that there is a normal cell, there could be a DNA damaging agent, which could be a chemical, radiations, viruses, and number of other things, and they induce some sort of a damage in the DNA. In normal circumstances, our body repairs those damages and makes the cell behave normally. But sometimes some mutations, some changes in the genetic material is so inherited that the DNA or is not getting repaired. And what happens? That leads to the mutation in the genome of the cell. This mutation may lead to three type of things. One could be the activation of the oncogenes. See, oncogenes are the genes which actually promote the oncogenesis. That means the carcinogenesis. So the first thing could be the activation or promotion of the oncogenes. That means the genes which are already available in our body, which carries the signals for the oncogenesis, carcinogenesis, they get activated. The second thing could be the alteration of the apoptosis, that the normal cell death process is affected and the cells are not able to die. And the third could be the inactivation of the cancer suppressor genes. So there are genes also which are actually suppressing the cancer mechanisms. All these three mechanisms are when they are altered, this leads to the expression of altered gene products and loss of regulatory gene products, and that leads to the formation of a malignant neoplasm. See, the problem is not when the cell has become cancerous. The problem starts after that. What does it affect? How does it affect the organ system or the whole body when a cell has become cancerous? Like suppose a cell has become cancerous and it is multiplying continuously. It is not death, apoptosis is not able to regulate that pathway. So what will happen? That cell will be consuming the energy. That cell will be requiring the ATP, oxygen, nutrients, micronutrients for their growth. And what will happen consequently? That the other cells will become deficient for the energy sources. Like in a normal cell, what happens? We all have learned that the glucose is a major source of energy. Through the glucose, through the glycolysis, the pyruvate is converted, is formed. That pyruvate is actually is converted into acetyl-CoA, which is starting material for the citric acid cycle and oxidative phosphorylation. Overall, total 34.23 ATP molecules are generated out of this normal glucose metabolism. But what happens? This oxidative phosphorylation and the citric acid cycle needs what it needs. It needs majorly oxygen for the formation of ATP. Whereas the glycolysis, you all have heard and read that glycolysis in an anaerobic respiration. That means it doesn't require too much of the oxygen. Now, since a tumor mass has formed, if you can see my hands, once a tumor mass has formed, it is growing and growing and growing and growing, expanding itself. Now, in the center of that tumor mass, in the core of that mass, blood vessels are deficient. ये एक ऐसा organ है, it is an organ system which is developing extra, which is not a part of the body, which is not required by the body. So what happens in the center of that core, center of that tumor core, there is a deficiency of the oxygen, and outside that center there is oxygen, but not a complete supply of oxygen. And the top layer of the cancer cells, it has a good supply of the oxygen. So overall, these three regions, we can classify them as epithelial cells. The one which is very close to the blood vessels. The another one is the oxphos cells. They have oxygen, but not sufficient oxygen for the normal functioning of the cells. And the core of the that tumor mass is hypoxic. It really doesn't have the oxygen. It is completely hypoxic in nature. As a consequence of this division of the cells, what happens that the epithelial cells, the outer layer of the tumor cells is has the sufficient amount of oxygen also, and other nutrients also. So there is a formation of uh, there is a glycolysis, and then there is the process of the citric acid cycle, and phosphorylation is completed very easily and very well. Whereas whenever we are coming down towards the oxphos cells, what happens that the glucose is available, but oxygen is deficient. So pyruvate is formed, but that pyruvate is converted into the lactate. Rather the pyruvate going into the TCA cycle or the oxidative phosphorylation, the pyruvate is converted into lactate, and this lactate is then further utilized for generation of the energy through the lactate dehydrogenase pathway. But the normal mechanism for the generation of ATP is altered, inhibited rather. Same way in the hypoxic cells, the situation become more worsen. Consequently, because the lactate concentration is increasing, pyruvate concentration is increasing, the cells become more acidic. Overall, in brief, if I'll conclude this part, it's a very complex phenomena, but in brief, if I'll conclude this process, what we can say that the hypoxic cells become highly acidic. Whenever the cell becomes highly acidic, it will die sooner or later. But to escape, since it is devoid of apoptotic mechanism, so it will not die. It will try to develop some other mechanisms through which it can survive and grow. And here only, from here, this hypoxic core only, the signals for the metastasis chips in. The signals for the metastatic the signals for the metastasis are released, and consequently metastasis starts. This is a brief about the angiogenesis and the metastasis. Metastasis is nothing. It starts with the process of angiogenesis. Angiogenesis is like whenever the core of the hypoxic cell, which is the core of a tumor mass, it faces a deficiency of nutrients and faces a deficiency of oxygen. It starts developing its own blood vessels, which is called as a angio. And genesis means the formation. So what happens that with the core of this hypoxic core, the signal starts, which helps them to generate the new blood vessels and invade into the nearby organs, so that they can extract blood through which they can get the oxygen and other nutrients. And whenever this angiogenesis is completed, whenever the new blood vessels are formed and they move into the nearby organs, or invade in fact into the nearby organs, then what happens that the signals for the cancers are moved from these blood vessels to the nearby organs, and this process is called as the metastasis. If I divide the types of cancer, we can broadly categorize them into the carcinomas, sarcomas, lymphomas, and melanomas. The carcinomas, which are basically the epithelial cell tissue-based carcinoma, which includes the breast cancer, lung cancer, skin and brain cancer, whereas the other side, that the sarcomas belongs to the connective and muscle, the cancers which are associated with the connective and muscular tissues, which includes the bone, muscle, cancers of the lymph nodes, and etc. The third type of the carcinoma is the leukemia, lymphomas, we call this, which it is not a solid tumor basically. It is a malignant growth of the leukocytes, and which leads to the excessive production of leukocytes. The fourth type is the melanoma. It is one of the most serious type of skin cancer which develops in the cells which is called as a melanocytes. So overall we can categorize the cancer in four categories: the carcinomas, which belongs to the epithelial tissues; the sarcomas, which belongs to the connective and muscular tissues. Blood is also a tissue, and in blood, whenever there is a excessive growth of blood, WBCs, malignant growth of WBCs, it leads to the lymphoma. And whenever there is uncontrolled division of the melanocytes within the skin, this is called as a melanoma. Overall, etiological agents which can induce cancers could be n number of. There are n number of substances which can induce cancer. But majority of the etiological agents are the can be chemical, physical, and the biological agents. As I have said in the initial slide, biological agents includes the bacteria, some parasites, viruses, whereas the physical agents includes the ultraviolet and ionizing radiations. And chemicals, number of chemicals are there, which could be the various components of the tobacco, the alcohol, arsenic, and other chemicals also. This is a brief about the various viruses which can be induce the cancer, but I will not get into it. As far as the treatment options for the cancer are concerned, depending upon the type of a cancer, maybe a breast cancer, which is a carcinoma, or maybe bone cancer or melanoma or sarcoma, various type of therapies are available. Moreover, the mode of therapy also depends upon the stage of the carcinoma. Like if suppose it is a benign based, so see, we can go easily with the surgery and remove it in majority of the cases. And if it has gone into the angiogenesis, or if it has metastasized, then we have number of other options which have very limited success. So now this was a brief about the cancer, and this was the first part of my presentation. The second part is new therapeutic options which have emerged in last decade, which is actually the small interfering RNAs, and in all the process is called as a RNA interference. But before I get into the RNA interference, well, let's recall the RNA. Let's recall the genetic materials available in our body, and then we will talk about the RNA interference. See, we all know that the DNA is the genetic material in humans, and apart from DNA, the genetic information is also transferred with the help of a RNA. RNA is a genetic material particularly in the eukaryotes, and RNA is basically a single strand structure of nucleic acid, and it they form the DNA and RNA. They both form or assemble as a chain of nucleotides, but the DNA is a double-stranded, as its the name suggests, whereas the RNA is the single-stranded, majorly single-stranded. Or earlier it was thought that it is a single-stranded, but later it has been observed that RNA could be a double-stranded also. That I'll be briefing you all in the next few slides. Biologically, RNA has been very defined, or whatsoever we have studied in our textbooks till now, that biologically RNA could be of a mRNA, messenger RNA, tRNA, rRNA, sRNAs. And apart from these four RNAs which I am talking about, they are actually the one which they contain some information. They contain information from one part of the message of the signaling pathway and carry that information to the other part of the signaling pathway. Whereas apart from these 4 RNAs, there could be other RNAs which we can call as a non-coding RNAs. They don't code for anything. They don't have any message for transmit to any other place. Like if I talk about the messenger RNA participates in the protein synthesis. And the same way, the tRNA, rRNA, and sRNA also participates in various mechanisms. Now, the other non-coding RNAs I'm talking about, they don't code for anything. They contain a self-complementary sequence that allows the part of the RNA to fold and pair itself into the double helix. These RNAs, non-coding RNAs, they are very short-lived. They have a self-complementary sequence which allows the RNA to fold and pair itself into the double helices. What it signifies, or what I'm trying to say, that these RNAs, non-coding RNAs, they have sequence which is self-complementary, and they can fold themselves or they can form themselves into a double helical structure. I'll be explaining it more in the next slides. Now, each nucleotide in the RNA contains a ribose sugar, as it is with the DNAs, and it has with carbon numbered from 1 prime to 5 prime. There is a base attached at the 1 prime position, which could be adenine, cytosine, guanine, and uracil. If I'll more strategically try to classify these RNAs, this could be a genetic RNA and non-genetic RNA. Genetic RNA could act as a genetic material like DNA, and they are single-stranded, usually, sometimes the double-stranded also. Non-genetic RNAs, they do not have, or they are not the genetic materials, and they are usually single-stranded, but they can actually be a self-complementary. Now, see, earlier it was very thoughtful that, okay, RNA has a very defined process in our body, and it will transfer one message in the genetic pathway from one step to the other steps. Later on, it was found that apart from these RNAs, some other short-lived single-stranded molecules are available in our body who plays specific role at specific point of a time. The only problem with these molecules is that they are very short-lived. They are formed and destroyed within few seconds. So at the end of the day, that's why these molecules were discovered quite later, because they were of a very small, they were very short-lived. So that is why they were discovered very later. But when they were discovered, it was found that, okay, these small short-lived RNA molecules have a very major role in the biological process, and by inhibiting or by affecting or by modulating the specific mRNA molecules. So what we have studied, that we have talked about the 4 type of RNAs, which were previously, which can be classified in 2 basic classes. But in these 4 type of RNAs, mRNA is a messenger RNA which actually coding for the proteins. Apart from the other RNAs, they can actually interact with the mRNA and destruct the mRNA. Consequently, what happens, that the signal which has been released by through the gene passed transported through with the help of a mRNA for the protein synthesis. When this mRNA is destructed, what happens, that the protein synthesis will not happen, and subsequently, or consequently rather, what will happen, that the signal for the biological signal is inhibited, and this is actually has been termed as the RNA interference. When these small RNAs were identified, it was found, these RNAs can actually affect the process of the mRNA formation, or they can degrade the mRNA, and protein synthesis is affected, and this process was termed as a RNA interference. And with the help of RNA interference, the whole phenomena was termed as a gene silencing, that we can silence one gene with the help of these small interfering RNAs. Now, how this whole process goes on and how this process can be exploited as a option in the therapeutic option in the cancer chemo prevention. So I will brief it about with the help of the RNA interference. See, there are three type of RNAs which can be used or exploited in the RNA interference. One could be the siRNAs. These are double-stranded RNAs with 21 to 25 nucleotide. The two nucleotides overhangs on the third prime region of the uh, uh, siRNA. On the other side, then the miRNA, if I talk about the miRNA, these are the single-stranded RNAs, which 19 to 25 nucleotide sequences. They are encoded by a non-protein coding genome. The third type of the RNA is the shRNA, which is called as a small hairpin-like RNAs. They are also double-stranded RNAs, which 15 to 20 nucleotides and contains a loop structure processed to siRNA. So that means these shRNAs are actually then later on processed to siRNA. They are formed into the siRNAs. The only thing is that they are structurally different from the siRNAs because they have a hairpin-like structure. I'll be showing how the structure looks like actually. The another important component for the RNA interference is the risk, which is called as a RNA-induced silencing complex. In very easily form, it is termed as a risk complex. It is a multi-protein complex which comprises of around 500 kilodalton proteins, and it incorporates one strand of a small interfering RNA or microRNA. This is the complex wherein the single strand, whatsoever the RNA, it could be the siRNA, miRNA, or shRNA, they will be converted into a single stranded to act in the RNA interference. So what happens that any of these RNAs, they interact with the risk complex to degrade the mRNA. So this is a basic typical structure of siRNA. As I have said in the previous slide, it is a double-stranded siRNA. It has 20 to 25 nucleotide fragments, which binds complementary portion of to target mRNA. That means that the portion of this siRNA is complementary with the mRNA. That is why it can degrade the mRNA. A single base pair difference between the siRNA template and target mRNA is enough to block the process. That means what is written over here is that even if the single base pair difference is available with the siRNA and mRNA, that would be sufficient enough to block or inhibit the mRNA. As I have said in the previous slides, there is a 2 overhang at 3' and 5' both. So we can see there is an overhang at 3'. And this is the process for the RNA interference. This is the whole process. It goes. This is a double-stranded RNA. This double-stranded RNA is cleaved, broken, into the siRNA of siRNA of 21 to 22 nucleotides. Dicer is basically a enzyme which is endoribonuclease enzyme belonging to the helicase family. It is RNase 3, and it cleaves the double bonds of the RNA to form a siRNA. Once this siRNA is formed, what happens that this siRNA interacts with the RISC. It forms a complex with the RISC, and this is called as an activated RISC complex, which leads to the unwinding of siRNA. Once this siRNA, the double-stranded siRNA, is cleaved, so what happens that one strand of the siRNA moves with the RISC complex, the other strand of the siRNA is left alone. and this is called as a sense strand. This sense strand is then degraded. It is of no use. And finally, what is left out? That is one single-stranded siRNA with a risk complex. What happens that this risk complex then interacts with the messenger RNA. The messenger RNA, which is actually coding or carrying the message for the protein synthesis. And the involvement of the messenger RNA, siRNA, and the mRNA leads to the degradation of the mRNA, and subsequently the translation process of the protein synthesis process is inhibited. When we talk about the microRNAs, they have a slightly different mechanism of action in comparison to the siRNAs. These microRNAs are the small non-coding RNA molecules, as I have said earlier, and they are found in plants, animals, and viruses also. They function in RNA silencing and post-transcription regulation of the gene expression. miRNAs, they basically are single-stranded RNAs, so they don't have that potential of forming the risk complex, but they act in an entirely different manner. What happens that whenever the miRNA binds with the complementary sequence, it's of the mRNA molecules, the mRNA is getting silenced. The major enzyme which is involved in this process is the DROSHA. DROSHA basically cleaves the helps in the formation of the miRNA with the pri-miRNA, which actually then forms the pre-miRNA and then leading to the formation of the mature miRNA. This mature miRNA then binds complementarily with the risk complex and leads to the degradation of the mRNA and subsequently inhibiting the translation. This is more brief about the pri and pre-miRNAs. Skip it. Then the third type of the RNA interference can be induced with the help of shRNAs. As I have said that it is a hairpin-like structure. You can see it is very much a hairpin-like structure. It is a short hairpin, which is a double-stranded 15 to 20 nucleotide sequence. It has a loop structure that is processed to siRNA. So shRNA is basically then converted into the siRNA. So after the formation of siRNA from shRNA, the process of the degradation of process of the inhibition of the translation is similar to what the siRNA, what is in the process of siRNA. The shRNA is typically expressed or typically the delivery of the shRNA is done with the help of the plasmids or through the viral or bacterial vectors, sometimes through the help of the electrophoresis also. shRNAs, they have advantage as a mediator of RNAi in that they have a very low rate of degradation and turnover because they are comparatively more stable in the physiological system, as siRNA is being a double-stranded and open-ended, it can very easily get degraded. But shRNA, since it is a loop structure, it is a more compact structure, so it stays within the physiological system for a more duration of a time in comparison to the siRNA, and that is why it has a better turnover in comparison to the shRNA. In fact, I must say that the shRNA induced silencing is more stable, and it remains there for a long duration of time. Both siRNA and shRNA use common molecules in their pathway, that is the dicer risk complex, and their major target is for the degradation of mRNA. So this is the mechanism of RNA interference in case of the shRNA. It is similar. The only thing is that this shRNA is then processed by dicer enzyme to form a siRNA, and then after everything is similar to what is done by the siRNA. See, it seems to be a very good strategy. It seems to be a very better way that how uh, that we can take one siRNA or we can take one shRNA and whatsoever the gene is available or promoting the carcinogenesis, we can easily cut it down and we can treat the cancer. But see, for every therapeutic regime or every uh, therapeutic process, there are some advantages and there are some disadvantages also. If I'll talk about what could be the possible advantages, that okay, it is a very highly efficacious potential mechanism for inducing the silencing even at the advanced stage of growth. That means if the cancer has gone into the metastatic stage and we can cut down, okay, these are the genes which are available over there and they are actually responsible for the metastasis. We can easily cut down those genes. We can cut down the translation process of those genes and we can cut down the metastasis. Second thing could be that this process could be highly specific, very specific, because we are targeting a single gene. We know that okay, this is the mRNA, the sequence of the mRNA which is to be targeted. So we can actually design a specific siRNA or shRNA which could be complementary to the specific mRNA and inhibit the whole process of translation. It has a precise functional mechanism, as I have said. It's a continuation of the second sentence I have written. We can target multiple genes of various cellular pathways involved in the tumor progression. So what we can do, that if we are finding it out, there are 5, 6, 7, 8, 9, 10 genes are involved, okay, in a particular tumor pathway. We can actually design 5, 6, 7, 8, 9, 10, 12, 20 siRNAs, and we can individually target those genes and subsequently cut down. It could be a future medicine for the it could be the future way out for the for developing or delivering the personalized drug for the specific patient. But there are some defects, there are some disadvantages. Sorry, there are some disadvantages associated with this also, that there could be structural defects in the siRNAs. Like whenever we have synthesized a siRNA or a shRNA is slightly more stable, but even then also there could be some defects. So there could be some defects in the siRNAs once we are synthesizing them. They are very vulnerable. They are very short-lived, as I have been telling in previous slides. They are very short-lived. Very easily they can be degraded. Why they are short-lived? Because they are unstable in the serum and they can be easily degraded by the nucleases enzymes. They can be very easily engulfed by the RES system, which is made up of the phagocytes, because the RES, the reticuloendothelial system, recognize them as the foreign particles. Since the size of these siRNAs is very small, they can be very easily filtered through the renal filtrations, and they are moved out of the kidney. So consequently, their half-life in the body is very small. On the other side, the other barriers, the other problems which with the siRNAs could be are that the tumor capillaries are very discontinuous. Sometimes they have very large openings and sometimes they have very small openings, which actually is conducive for the transmembrane transport of siRNAs in tumor tissues if it is actually a large opening. Then cell membrane is a negatively charged structure, and siRNA is also a negatively charged structure and is water soluble also. So therefore, we need to devise some mechanism through which we can transport these siRNAs into the cell, because both are negatively charged. So we have to use some sort of a mechanism so that we can transfer these siRNAs into the cell. There are a number of ways. Sometimes some lipids are used. We can encapsulate these siRNAs in the lipids and transfer them into the cells. Or some viruses are also used. But sometimes we incorporate these siRNAs into the viruses, and then we ask those viruses to get into the cells so that the virus can replicate and deliver that siRNA into the cell. Nowadays, the electrophoresis is also being used for the delivery of these siRNAs. One more thing is that once this siRNA reaches the target cell, it is encapsulated by the early endosomes for the transport and fusion with late endosomes. So there are endosomes nearby the cell sites, and what happens that whenever the siRNAs reaches to the cells or nearby to the cells, it is actually easily transported with the help of endosomes. Two other major issues with the siRNA therapy, or which needs to be overcome, are since they act as a foreign material to our body, they can stimulate the innate immune system of our body. And one big problem which currently majority of the scientific scientists are facing across the globe is that to reduce the off-target effects, since it is nothing but a sequence of 15 to 20 nucleotides, there could be a possibility that out of those 15 to 20 nucleotides, uh, that sequence may be complementary with the other mRNA sequence. Subsequently, what may happen that we have gone to affect one particular mRNA and lost the or affected the other RNA. It is a similar way that a drug has a side effect or a adverse effect. This is my small team we are working with. Thank you. That's all from my end, Dr. Sanjay.
Yes, thank you so much, sir, for this wonderful session. And we have got few questions from the chat box from the audience, sir. Sir, first one is are there any clinical trials which involves the siRNAs for the cancer treatment? And what are the anticipated outcomes from that clinical trials?
Of course, there are clinical trials. Of course, there are clinical trials who failed. Okay.
Okay.
As far as the outcome is concerned, the outcome is we anticipate a, a formulation which have the which we can utilize in the metastatic cancer.
Okay, sir. And sir, I have one question. So, can you share any insights into the extramural grants that you have secured from your research and their impact on advancing cancer chemoprevention?
Sorry, I'm not getting your point.
Sir, can you share the insights into the extramural grants that you have secured in your group and that are impacting the advancing cancer chemoprevention?
Mother, you want to say that what are the grants we have received?
Yes. And what are their
what are see we have received majorly 4 grants. There are small grants also, but we have received major 4 grants. Out of the 4 grants, the 2 grants were working on the area of, not 5 grants right now actually, if I'll say. The latest grant which we have is from the Department of Health Research, wherein we have received funding for the development of the small interfering RNAs, to target the MCT1 and MCT4. MCT1 and MCT4 are the channels. I'll show you just. Okay. See, here is the if my slide is visible, then you can see there is the MCT4 channel, okay, and then on the normal cells which are not actually the part of cancerous mechanisms, they have a MCT1 channel. What happens whenever there is anaerobic mechanism, lack of oxygen based mechanism, there is an increased formation of lactate, and this lactate makes the environment acidic. And since the environment is acidic cell will die automatically. But what happens that this lactate is transported back from cancerous cells to the outside the extracellular domain, and then it moves to the normal cells. So what we have secured, what we are doing right now, that we are devising the siRNA, designing the siRNAs for the MCT1 and MCT4. What we are trying to do out of this is that if we are going to inhibit the translation of the MCT4 channels and MCT1 channels in the cancer cells and normal cells both, what is going to happen, that there will be an accumulation of the lactate. Once the lactate is accumulated in the cells, this hypoxic cell is going to die by its own. We don't need to do anything extra. The best part of this strategy is that the MCT4 cells are only expressed when there is a deficiency of oxygen. ये कहाँ पे होगा? It will happen when, like, either in the cancer cells, or sometimes when we go to gymming, the boys go, or the girls go for a gymming on the first or second day of a gymming, they do excessive exercise, rigorous exercise. So what happens when they come back, they say that there is a pain, because what we say, lactic acidosis, lactic acid deposition is there. Since we have done a rigorous exercise, which our muscles were actually not used to, and suddenly we needed a good amount of oxygen and ATP in those muscles. So what has happened? Because suddenly and immediately the cells will not start doing those requiring or meeting out the requirement of the oxygen. So what will happen? There will be an excessive lactate production. So how our strategy is advantageous, that these type of cells where the MCT4 is expressed will be only in two situations. One would be in the cancer cells and another would be in the exercise. So I don't expect any cancer patient to go and do gymming. So that is why I feel that, okay, this could be one of the way out. Another thing is that the another extramural grant which we are working with is that this hypoxic cell. These hypoxic cells are regulated with the help of an enzyme which is called as a protein which is called as a hypoxia inducible factor 1. You can see, I'll enlarge the screen. Hypoxia inducible factor 1, HIF1 alpha. This HIF1 alpha is normally present in our body. It is always present in our body. But it usually degrades automatically with the help of the enzyme which is called as a prolyl hydroxylase 2 and factor inhibiting hypoxia, FIH1. These are the two enzymes which actually in normal condition, they are degrading the HIF1 alpha protein with the help of a proteasomal degradation, with the help of 2-oxoglutarate, oxygen. But once the cell becomes hypoxic, there is no oxygen, the degradation of HIF is actually affected or lost. And this HIF actually generates the signals for the metastasis. So another way out is that we are targeting the prolyl hydroxylase 2 and factor inhibiting 1 alpha. Earlier, I think we got one grant in 2017, wherein we developed some small chemical entities against the prolyl hydroxylase 2, or for the prolyl hydroxylase 2. And recently we got the grant for developing small chemical entities for the factor inhibiting 1. So these are the two major areas which are actually currently we working with. One is with the MCT4, wherein we are developing the siRNAs for the MCT4 and MCT1, whereas we have developed several small chemical molecules for activating the FIH and PHD2.
Thank you so much, sir. Let's move toward the concluding remarks. In conclusion, today's webinar on SiRNAs, a new era in cancer chemoprevention, has been an enlightening journey into the forefront of pharmaceutical sciences. We extend our sincere gratitude to Professor Gaurav Kaithwas, sir, for sharing his profound insights and expertise in the field of cancer biology. The collective knowledge and discussion that have transpired here today underscore the transformative potential of siRNAs in revolutionizing cancer chemoprevention. As we move forward, let us carry forth this newfound understanding, fostering collaboration and innovation in our ongoing quest to combat and prevent cancer. Thank you to our esteemed speaker, participants, and the dedicated team behind the scenes for making this webinar a resounding success. Together, we take strides toward a future where siRNAs play a pivotal role in shaping a healthier world. We extend our sincere gratitude to Dr. Shashi Alok sir, National President of SPSR, for gracing us with this esteemed presence today. In acknowledging your dedication and leadership, we express our heartfelt thanks for your invaluable contributions. In extending our deepest gratitude, we express our sincere appreciation to the esteemed Professor Gaurav Kaithwas sir, whose generosity in sharing his profound expertise and invaluable perspective has enriched our own understanding of SiRNAs in cancer prevention. A special acknowledgement is extended to our convener, Mrs. Monica Sabarwal ma'am, whose pivotal role in orchestrating this insightful event has been instrumental. We extend heartfelt thanks to all participants, recognizing the collective effort that has effectively pushed the boundaries of knowledge and fostered a spirit of collaborative learning together. Your contributions have made this webinar a resounding success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. Thank you for being an integral part of 38th SPSR webinar. Your active participation and contribution have been invaluable, and we look forward to future endeavors that contribute to the continuous betterment of healthcare and well-being for all. Have a wonderful day. Thank you. Thank you so much, sir.
Thank you. I can leave now, na?
Yes, sir.
Okay, thank you.
Thank you so much, sir. Thank you.